In this video, we will discuss about the effect of this NPY protein on the pancreatic cancer or pancreatic adenocarcinoma. NPY protein is known to affect or highly upregulated in many cancers. So researchers wanted to find out whether this NPY protein has a similar effect on the pancreatic adenocarcinoma or not. So in order to do that, they took two mice model. Uh, One has low metastatic mice model, another is high metastatic mice model. The difference between low metastatic and high metastatic mice model is that the low metastatic mice model has the loss of p53 functions, and this high metastatic mice model has the gain of p53 function. So p53 genes is upregulated and p53 genes is downregulated in the low metastatic model. And then they check the NPY proteins in these two models, and what they are able to find out and using the qPCR, the relative mRNA levels, and what they are able to find out is that the results in the panel B is not about this high metastatic and low metastatic. It is about the mice with has pancreatic cancer and the normal pancreas, and it shows that the mice that has a pancreatic cancers has high levels of these NPY expressions compared to the normal pancreas, whereas its sister mRNA levels such as PYY and PPY are either statistically down regulated, as in the case of PYY. We can see that compared to the normal pancreas, in the pancreatic pancreas the PYY proteins is down regulated, or it is there is no change in the case of PPY. The difference, there is no statistically significant difference between pancreas PPY in normal pancreas and in the pancreatic cancer. And then they check the NPY proteins using the immunostaining in the normal pancreas, the pancreas with the low metastatic mice model and the pancreas with the high metastatic mice model, and they are able to see an increase in the NPY protein levels from normal pancreas to the low metastatic mice model pancreas, and it's highly upregulated in the highly metastatic pancreatic mouse models. And this quantification of this NPY proteins in this immunostaining is shown in the this right hand side of the panel C, where we can see that the highest NPY positive cells are found in the highly metastatic mice models. And in the case of highly metastatic mice model, they also check the NPY protein expressions in the liver, and they say that in the liver also the NPY protein expressions are found. Thereafter, they check the NPY proteins and its receptor, whether they are co-expressed or not. And what they are able to find out is that in the case of normal pancreas, the amount of NPY and NPYR are very lower, almost non-significant. Whereas in the case of primary tumors in the low metastatic mice model, they are able to see that both NPY and NPYR are coexist, indicating that they exist close to each other, or we can say that in the same cancer cells. And the amount of NPY and NPYR are higher in the highly metastatic models compared to the low metastatic models. Thereafter, they check this NPY and NPYR using the pancreatic cancer markers, that is keratin 19, and they are able to see that both this NPY is co-expressed wherever they find these keratin 19 markers, indicating that NPY is expressed on the cancer cells. And similarly, they also see that NPYR is also find out wherever they find the KR19 markers, and this also indicates that NPYR is also expressed on the pancreatic cancer cells. And these things they have find out for the primary tumors in the low metastatic mice models, and also in the high metastatic mice model, and even in the liver metastasis of the high metastatic mice models, the similar observations has been found out, indicating that this NPY proteins maintain its high expressions even during the metastasis. And these results are on the mice models. So now they wanted to check whether similar phenomena or similar observations are there in the human patients or not. So they use the cancer genomic atlas, and here they check the pancreatic cancer cell patients with the NPY expressions, and they're able to see that compared to the normal pancreas, in the pancreatic cancer affected patients has the high NPY levels. And when they check the NPY receptors compared to its other similar receptors, they're able to see that NPY1R receptors are significantly higher in the pancreatic cancer patients compared to the pancreatic cancer patient samples compared to the other similar receptors. So that is NPY2R, NPY4R, NPY5R. And thereafter, once verifying the data in the cancer genomic atlas, they take the human patients and check the NPY expressions in the adjacent normal pancreas to the human pancreatic adenocarcinomas, and they are able to see that NPY expressions is statistically higher in the pancreatic adenocarcinoma compared to the adjacent normal pancreas. So all this data indicates that there is a high expression of NPY proteins similar to what people have observed in the other cancers, and from there they made a hypothesis as if NPY proteins is downregulated, then it might suppress the metastasis, some of the metastasis in the pancreatic cancers. So we will discuss how they validate those or check that hypothesis in the subsequent videos. So far, if you need to understand more about their experimental results that are presented in this figure, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.